Thank you for this opportunity. I particularly like to thank Professor Thomas Carroll, who invited me to be part of this August gathering. And I am delighted to be in front of such wonderful, wonderful audiences. And again, thank you so much for this, to honor my beloved Professor Albert Eschenmoser. So I start with the slide, and it's really important to really read through this. So when he was interviewed at Scripps regarding what he thinks about origins of life, he said the crux of the question of origin of life lies in the fact that it refers to the chemical processes that happened billions of years ago. The origin of life cannot be discovered. As other things in science, it can be only reinvented. He said reinvented because life has already been invented once. And most importantly, he also said, in contrast to what some people may think, you cannot be overly ambitious. And that is what this field kind of suffers from at this moment. There are a lot of claims that are made, sensational things in the newspapers, as you would see, only to be rolled back within a couple of years later. There'll be, he said, the absolute you can really dream of is a chemical model. And that's what many of the practitioners in this field are trying to do. And in such that any effort is worthwhile and important. So all the talks that you will learn, hear in this session and in the afternoon session are important because people are trying to create an understanding for what is the chemical process that was involved in the origins of life. So for me, I look at it as a problem of transitions. And what I mean is the following. So I think most of us are comfortable with this picture in this room. And so, no. So, but, this is possible as, we heard in the earlier talks that there are these fossils that you can lay your handle on, and then people can, go back with many of these things to do this, because you can go back. But the problem is once you take this away and replace this with molecules, then it becomes really tough, because there are no chemical fossils that have lasted. As we heard from the talk yesterday, that there has been so much of diagenesis and recycling that you do not know that if you find something 3 billion years ago, is it now a product of life from today, or was it a product of life from 3 billion years ago? So there is always this doubt. And there is always this trouble of going back and forth. And as you all know very well, we just can't mix chemicals in a beaker and hope for life to climb out of it. No, that thing has not yet happened. And the black boxes here refer to not to the inability of us to do the demonstration. We can do such a demonstration of converting one to the other, but your question will always be, how do you know whether it happened like this 4 billion years ago? And there's no way of answering it in a very definitive manner. So the question is, how can we, or what can we know about the chemical origins of life? And one good place is to start with what life as we know it, and then work our way backwards, and that's what many people do. Before that, I just want to introduce this camel problem. Please don't Google it. Some of you may know the answer, but just bear with me, and I'll show you what the relationship is this with respect to what I'm going to present. So, father, 3 sons, 17 camels, they open up the will, and this is the math he gives them. So everybody should be getting this much of camels, and if you look at the math, you're not going to have a happy camel or a happy family, okay, because it's going to be difficult. So how do you solve this problem? We'll come back to this, hold this in your thought. So going back to using life as a guide for origins of life, there is the famous molecule RNA that many people focus on. They look at the constituents. Here I'm just focusing on the pyrimidine nucleosides. And people look at what sort of simple chemistry that can come from simple molecules that could have existed on earlier, like hydrogen cyanide. So hydrogen cyanide will polymerize and give you these kind of bases, and you immediately see these beautiful, let me go back. Oh. So I was here. So they focus on these kind of molecules, and then they say, okay, you see the same type of structures that you can produce by the simple chemistry, and therefore you have that. And therefore they try to make the connection But if you look at biology of how it makes it, it does not synthesize these nucleobases, but it starts from what is known as orotidine phosphate. And it decarboxylates to convert it to uridine, and then aminates it to produce the cytidine. And if you look at where this orotic acid, that comes from, it comes from orotic acid, and biology produces orotic acid from an amino acid called aspartate. So there is really no HCN chemistry there. But people tend to focus on that. But what is really remarkable is the same chemistry that gives rise to the canonical nucleobases also gives rise to this orotic acid. But nobody focuses on the orotic acid. They only focus on cytosine and uracil because that's what the end product RNA has. And it's important to note that orotic acid is the only nucleobase that we know so far that is also synthesized in a prebiotic fashion, and biology synthesizes it. The 4 nucleobases that we always think about, AUGC or ATGC, are not synthesized by biology. They are built atom by atom, especially the purine synthesis. So if you look at nature as a chemist and you want to employ nature for making your nucleotides, you would fire it in a minute in your company. You would not hire that, because the way it makes it, it's, as Hochberg clearly put it, in a very deterministic fashion. So why people do not focus on orotic acid is because it's not in the final product RNA, so they don't worry about it. So we ask the question, what happens if we take orotic acid and put it in an RNA? What happens to it? How bad it is? So what you find is, so these are what we call UVTM figures, which show how strong the base pairing is when you bring in 2 duplexes. The black line that you see is of the natural system RNA, and you see it's quite strong. So the more the curve is to the left-hand side there, it's much stronger. And you see the red curve is one substitution by the orotic acid, the O, and you see it completely destroys the base pairing. So when I took this to a colleague of mine at Scripps and showed it to him, he said, oh, this is not surprising. I said, why? Oh, nature just chose a better base pairing partner, and that's it. And the question was, why is it so bad? It's bad because the carboxyl group at the 6th position of orotate interferes when you want the Watson-Crick to face the right way for base pairing. And if you substitute it with an NH2, it's not the negative charge, it's just the pure steric clash that happens when you want to base pair. So the question is, when did nature know that orotic acid is bad and uracil is better? You had to wait till you had a complementary base pairing partner, and it's only at the level of an oligomer you can make the decision. Not at the level of a monomer, not at the level of a dimer. So even you say, oh, I found nucleobases in the meteorite, that's good to get excited about the building blocks of life, but that doesn't mean anything for evolution in terms of how you can bring it back in terms of functional capability at the level of an oligomer. The other part is, coming to the meteorites give you a slew of compounds. All of them have materials that are considered to be building blocks of life. And the people get excited over this, and they begin to say, oh, building blocks of life. But what you have to keep in mind is it's one among many thousands of compounds. It's very difficult. So what it means to the origins of life is that the chemistry is universal. And what it is like this. This is the face, a formation on Mars, which looks like a face. And, we get excited about that. But in the popular press, what happens is it has a life of its own. And this is what happens, even to many of us who are working in this field. Building blocks of life is not life. They are just building blocks of life. That's it. So if you want to know how the actual face looks like, that's how it looks like. So we are fooled by the connection between biology and what we think to be in prebiotic chemistry. So there is the prebiotic chemistry. There is biology. We are trying to work our way up or work our way down, and unfortunately, we don't meet. There are some experiments where I say, oh, maybe I would change a little bit of thing. Maybe I can fit one part, but then the other parts go out of alignment. And what I got from Google when I was looking for a picture was more like this. That is, there is really no continuity from start to finish in the whole process, but a semblance of continuity that is there. And I will tell you with concrete examples. So now the most interesting part of the talk, the answer. So when you do not know the answer, you go to a wise person, and then they say, okay, well, he says, I'll bring a camel. He brings a camel and then adds to it and does the math, and it adds up. And now if you add the 9, 6, and 2, that comes to 17, and he takes his camel back. Okay? Are there molecules and processes that happened early on the Earth that happened, made the transition possible, but are not in life today? And that's what we are interested in trying to find out. So how does that 18th camel help in research? I've given you this philosophy, aptly following a philosopher. So that's where our beloved professor comes in. He asked the question in terms of not how RNA, but why RNA? Why these molecules? Why did nature choose these molecules? So he chose, he said, if these are the molecules that could have been available, how did nature settle on these two type of molecules? And he started making alternatives. So these are the sugars that are made from hexoses. So you can ask yourself, and this is a famous thing, game you can play. When the moment the word sugar is mentioned, what is the first thing that comes to mind? People say sucrose, fructose, glucose. Rarely people say ribose. Okay? Ribose is not even considered a sugar in many people's mind because it's part of an RNA. Okay? So when he did this, he found that all these hexose sugars gave very weak base pairing. And so now you can say, oh, nature chose a better base pairing system. But wait. He then did what is known as an isomer of RNA, where instead of a furanose 5-membered ring, he switched it to a 6-membered ring. And all of these were much better base pairing than RNA. And the question, of course, was, did nature miss a better base pairing system? And his answer was no. He said, if that is stronger and this is weak, it's like the Goldilocks, too hot, too cold, just right. And he said that RNA must be just right because it is by optimization. So evolution is more by optimization rather than maximization of base pairing strength. But optimization with respect to what? And I would argue that it's with respect to the environment in which the molecule is made and is allowed to function. I'll show you that. So when you go and look at the literature, you can see that RNA 4 nucleobases are compatible with almost all backbone systems, many backbone systems in the antisense industry and things like that, biology and medicinal chemistry. But if you take alternative to the bases, there seems to be very few by comparison. There are only very few alternative bases that can function with respect to the backbone from a prebiotic perspective. Benner has created a lot more bases that are more important from the biological perspective. So why is that so? That is because of the pK of the nucleobase and the pH of the medium, which are referring to the ability of that molecule to accept a proton or donate a proton in the solvent that is in. And for us, the solvent is water. If you assume a pH of 7, which is what our body is around that value, you will see that there is a gray area in which there are molecules that have protonated or deprotonated, so they are charged. And what happens is they begin to interact with the water molecules, and thus they are not able to interact with their base pairing partner. What happens is the bases, the nucleobases by themselves, are quite hydrophobic. They are not protonated or deprotonated at the physiological pH, and therefore they are able to interact with themselves. It's like two oil droplets finding themselves in water and sticking together. So if you really put this in an ancient lake, if you will, of a pH of 7, and you start looking at all the nucleobases that are available throughout the literature, you see there is a kind of an hydrophobic dry land on which our canonical nucleobases sit. And this has important implications because this is at a pH of 7. And what it tells you is when nature decided to go with these kind of nucleobases, the environment must have been around pH 7. If this lake had been at pH 4 or at pH 11, you would have selected entirely different sets of nucleobases. So that's the optimum range when the aqueous medium is pH 7. So now coming to this question of how do we go about understanding the structure function of RNA? The question that we asked based on Eschenmoser's work was, why not pentulose nucleic acids? These are, again, isomers of RNA. The ribose and the 4 sugars that I've shown on the left-hand side are what is known as aldoses. They have the aldehydic carbonyl group. The ones that are shown in blue are the ketoses, which are the keto version, where the carbonyl group is now moved one into the ring. And they also look like RNA when you draw them like this. And the question was, we asked, just like Eschenmoser did, why a pentose and not a pentulose? What is wrong with this kind of a sugar? So we started making these oligonucleotides. And here are the 3 different structures. And in relation to what RNA looks like, you can immediately see they are very different. One thing important is that the red phosphate backbone that you see is now on the same side as the nucleobase. So that means and if you look at RNA, you can see the green bases and the green phosphates are far away apart as they can be. And that's the beauty of that. So when we did this, the 3 postdocs worked on this very hard. We found nothing. There was no base pairing, nothing, and so we had to write a paper out of this. So whenever you get these kind of negative results, you turn philosophical, okay? And you write a nice philosophy paper out of this also. So we said, if these were competitors to RNA, they would not have functioned. That's quite obvious. But more important was that being abundant is not that you will survive by selection. And I'm not talking about many colleagues in our office, but being there and being useful are 2 different things. Okay? That is the important message that we got from this. Now, what is the importance of the ribose? So the ribose ring, that's how we started this one. So we made another acyclic backbone, which is derived from glycerol, a 3-carbon polyalcohol. And we compared it to another molecule that was proposed by Leslie Orgel, Jerry Joyce, Schwartz, and Miller. And it was also studied by Stephen Benner, who showed that this is called the flexible nucleic acid, where they have removed one CHOH from RNA. And it has poor base pairing properties. So when we compared it to our system, we saw that they both have the same backbone. Only the linker connecting the nucleobase and the backbone is different. Why this difference in base pairing? And this beautifully connected to our pentulose system that I showed you before, and I will show you, I'll go through this a little slowly so that you can watch this happening. So there is RNA, and I have highlighted the red ring structure, and this is what Leslie Orgel, Schwartz, and Miller, and Joyce removed to get to this flexible nucleic acid. Okay, so that we remove the red portion. Now what happens is you turned a good base pairing system into a weak or a poor base pairing system by removal of the ring. Now I'm going to do the same thing to my pentulose nucleic acid, and if you remember, this had no base pairing, nothing whatsoever. I'm going to remove the ring now, and if you remove the ring, you go to our iso-GNA, which has much better base pairing. So the same operation leads to completely 2 different results. Why? If you look very closely and you look at that arrow that I pointed out, that arrow is important for the Watson-Crick phase facing the right way. So what happens is, if I remove the ring in RNA, I create this very flexible system that causes problem for the Watson-Crick to face the right way. But in my original, the pentulose system at the bottom, it was already facing the wrong way. By removing the ring, I allowed it to face the right way, and now I regain my base pairing. And so now, that long question in my mind, what is the function of that ribose as a cycle, came to fore, which means that that ribose ring structure is responsible for directing the base the correct way for the Watson-Crick to function, and therefore your base pairing is there. So the ribose ring is not just only for giving this inflexibility to that molecule, it is also orienting the base in a proper way so that the Watson-Crick faces and makes the base pairing. So with all this, you look at RNA, it's a wonderful molecule. It has been optimized, each and every part of it. I won't go through the list, but you can see the sugar has to be a pentafuranose. It can't be anything else. It has to be a ribose in the furanosal form. The nucleobases have to be in the beta form. They have to be in the 3' 5' linkage. You have to have the bases correct with the correct pKa, or else they will not base pair. So all this points to this optimization of this molecule over a period of time. Also, many properties emerge only at the level of a polymer, not at the level of a monomer. So you can't talk about base pairing, Watson-Crick, Hookstein, selectivity, information at the level of a monomer. It simply doesn't exist. They do not base pair as a monomer. You have to get to an 8-mer or a 12-mer before this emerges and manifests itself. And therefore, our take is that RNA is fully manifested as a biopolymer only as a polymer and only at the aqueous neutral medium, neutral environment. And this indicates it's a highly, highly optimized molecule for a given environment. And therefore, we may consider RNA to be a product of chemical evolution and selection. What does this mean? In the context of how you go from a mixture, so that means, let us say, one of you has been selected by NASA. You are the lucky person to go to and land on an exoplanet. And you land on an exoplanet and you find the set of chemicals. Can you already predict what life should be based on, on these molecules? You may have a bias based on our RNA, DNA, both protein-based life, but you need to consider what the molecular set is. Do you select a sugar as a furanose or a pyranose at this level? You do not know. Let's go to the next level. Let us say you have added molecules to that. Now can you make a decision as to what life should be based on? Probably not. You have another level, another level. You really can't decide until a function emerges that you can relate to as what you would call related to life. So you can have different levels of selection, stability. For example, Dimitar will talk about stability with respect to UV radiation. You can have solubility issues. You can have molecules that are soluble, stable, et cetera, et cetera. But you can't decide on a property that has still not emerged. Because if you try to do that, then you are playing the role of a teleological evolution that you cannot do. And as Francois beautifully said, evolution is a tinkerer, not an engineer. That is also true for chemical evolution. You can't already impose a purpose on prebiotic evolution and say, therefore RNA, therefore proteins. That's not possible. And this is what I call blinded by biology syndrome in this field. People are blinded by biology. They ram it through all the way to prebiotic chemistry where there is no biology. And there was a famous quote by Nick Lane who said, it will be foolish to ignore life as a guide. And my response to that, yes, it is foolish to, it would be foolish, but it's even more foolish to use it as the only guide. You cannot do that, and you should not do that. So how do you go from a mixture to a complete homogeneous system? And the type of research program that we have is to say, let the mixture, under a defined conditions, form certain things. Let that property emerge, and perhaps that would lead to these kind of emergence of this. And I'll give you a concrete example. So far, I have been speaking in a philosophical, hypothetical term. Where is the actual, as you would say in the West, meat on the bone, right? So let us start with peptides. So many people focus on amino acids because that's what peptides are made of. So you get excited when the Urey Miller gives you amino acids, meteorites give you amino acids, therefore proteins. But it's not so fast. It's because it's quite debatable, and people are still trying to find good solutions to that. But if you look in the context of prebiotic chemistry and look at the very famous Urey Miller experiment, this is what he became famous for in Science and New York Times and everywhere. But actually, if you go and look at the experiment and read through it very carefully, you also produce a lot of hydroxy acids. But they were ignored. Imagine if Miller had published, I had found hydroxy acid, Science would have not published it. And he would certainly be not on the New York Times. So you look at the meteorites, it gives you more hydroxy acids than amino acids. Why are we ignoring the amino acids when prebiotic chemistry gives you both? The answer is simple. Because biology does not use alpha hydroxy acid as a polymer. That's the blinded by biology syndrome. And so the question that we asked was, if we mix those two, can we find a solution, a potential solution to peptide formation? Yes. So what you do is you take the hydroxy acids and amino acids, and you just dry it down. No catalyst, nothing. Just dry it down. Add water again, dry it down. And what happens is, with the hydroxy acids, as you know very well, they form these polyesters, the polymers that you are wearing. The polyesters are formed very easily because it's a very kinetically favorable process. But once you make that polyester, that gray bond that is circle, that becomes activated to attack by an amino group. So we attack it by an amino acid which exists there, and now you see you have a free acid again, the blue acid. So you make another ester by just wet dry chemistry. You make another amide. You continue. And before you know it, before you want it or not, Amides and polypeptides are a natural outcome of this simple wet dry cycle when you have a prebiotically plausible mixture of alpha hydroxy acids and alpha amino acids. And so that which was very difficult when you try to start only with pure amino acids, which is not possible, by the way, on prebiotic Earth, it becomes much easier, and it's a natural solution that comes out on its own without any imposed teleology. And what is so nice is that if you look at the ribosome of how it makes, it's exactly the same chemistry. Two esters are made, and the amino group attacks. I'm not claiming that we found an ancient mechanistic pathway. I'm just pointing out the coincidental between these kind of systems. One is highly sophisticated with sequence-coded information. The other is simply crude primordial chemistry. So the same type of chemistry that now you use, and now the question is your alpha hydroxy acid the 18th camel that was added and goes away? Added, goes away. And in chemistry parlance, we call it a catalyst. That's it. So now, can we translate this to nucleic acids? We can. I won't spend much time on the slide, but just to say that we can use the same principle of chimeric nucleic acids, building where you mix two different nucleic acids like RNA and DNA, make the chimeras, and those chimeras actually help you replicate RNA and DNA much better than the pure RNA and DNA sequence as well. So the implication is that you do not have to start with clean materials. You can start with a mixture. Because you start with a mixture, you have a possibility of getting two systems simultaneously. That means RNA and DNA could co-emerge side by side, and that could also lead to emergence of homochirality if you start with the D and L. So these mixtures actually give you a much better system. So now back to the same principle here, that you can have different building blocks, different chemistries, but this, are similar building blocks as well mixed in, and then you switch over to the homogeneous backbones and peptides. And so these are the chimeric sequences here would be the 18th camel that come and goes. And then, but then the question is, if you have many such variations, if there is a change in conditions, you can have a completely different paradigm. And what would those alternative paradigms look like? I'll give you an example. So as Albert Einstein famously said, we not only want to know how nature is and what all she does, but to know why nature is such and not otherwise. So what is that otherwise of nature? If you go and land on an alien planet, would we even know what life is? Because, leave alone the definition, should it move to be life? Should it do all the things that we do in order to consider it to be life? Just think, before we even knew the bacteria and viruses, life was considered to be at the level of plants and worms, right? So back to our system, the pentulose system, which, this is the data for the people who are interested, just to show that, if you insert one molecule of that into RNA, it completely destroys the base pairing of RNA. So for a different context, I asked one of my postdoc to say, why don't you make a completely alternate system? So you have a pentulose, pentose, pentulose, pentose, a mixed backbone, completely with 50% insertions, for a different reason. And after he did that, I asked, did you measure this? Is it base pairing? He said, no, it cannot. But what we did was, when he did measure, it had beautiful base pairing systems, and it's almost comparable to RNA. And we were shocked, because we were mixing a system that is not base pairing with RNA alternatively, and it made a system that is almost as good as RNA. So we quickly switched to ribulose system, and this is just to show the data. I won't go through this in detail, but just believe that you are able to make systems that, when you completely mix systems that don't talk to each other, you can still make it. And here is one example of this, where you make completely mixed alternating sequence. And it's not surprising now when you look back, just as you need complementarity fit with the nucleobases, you just need a complementarity fit with the backbones. That makes sense. And now the question is, with this, can we change the paradigm of Watson-Crick as we know it? And that would be, so now what we are doing is actually, somebody said, we are creating Frankenstein nucleic acids. We are just mixing and making different systems that can have 3 different building blocks and make nucleic acids out of them as well. So this is what we know generally. GC is stronger than AT, and that is known all throughout biology. Okay? Now, here, we have two different sugars and two different bases. So now I can play the game of which sugar will contain which base, the purine or the pyrimidine. Here the blue, which is the ribulose, contains the adenine purine, and the black, which is ribose, contains the thymine, which is the pyrimidine. And the same with the guanine cytosine. But now I can switch it, and now you see here is a system where AT is much stronger than GC. We have completely flipped the base pairing paradigm of nucleic acid chemistry. And this was very, very surprising to us. So now you can see that AT is stronger than GC. And now it's not that thing which interested me. But now if you look only at AT, I can now play the game that many people wanted to play, that if I take if I take the adenine on the pentulose and thymine on the ribose, I can get an AU pair. I just switch that, keeping the same sugar backbone, and now I get a GC base pair. So I can have only a dyad of pentulose ribose, but switch the same bases back and forth and create a completely different sequence. That means these keeping ATA in the same, but using the ribulose nucleic acid RNA backbone, I can create an RXNA that can completely change the type of base pairing paradigm that you know. And so, coming to the final slide, where you have this kind of system, the question is, are the molecules that we see today the same that you would get many times? And so the question is, if you change the temperature, malt solvent, et cetera, you probably will get to a different biological paradigm. And this is a message I want to leave you with, is that the molecules that we have in today are a destination and not a destiny. You change the conditions, you get a completely different system. With this, I thank the group. And also, I have to thank my professor, mentor, who introduced me to this field, Albert Eschenmoser, and to the people, young people who are coming to this field, irrespective of what was told to our Thomas Carell, that it's only for retired professors. Thanks to him, there are talented young professors in this field who are coming up, as you will see in this afternoon session. And I leave you with the slide and the thought. And once again, thank you so much for your kind attention, the invitation, and I'm really grateful for this opportunity. Thank you so much.